Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. In today's class, Embryology Foundation Module Lecture 7, we are going to discuss the process of the fertilization. So fertilization is the complex sequence of events that begins with the contact between a sperm and a secondary oocytes and ends with intermingling of the maternal and paternal chromosomes at metaphase of the first cleavage of the zygote. So first of all, before the process of fertilization starts, there must be conditioning of the sperm. Only the conditioned and the capacitated sperm can fertilize an oocyte. So sperm in the female genital tract before fertilization undergo process of capacitation and then the acrosome reaction. So capacitation starts in uterus and continues into the uterine tube, but most of the process of capacitation takes place in the uterine tube. It involves epithelial interactions between the sperm and the mucosal surface of the uterine tube. The follicular fluid which is present over here enhances the process, and this process takes place about 7 hours. So what happens in this process of capacitation is that the glycoprotein coat and the seminal plasma proteins are removed from the plasma membrane over the acrosome. So only the capacitated sperms show as they show no morphological changes, but they are more active. Completion of the capacitation permits the acrosomal reaction to take place. So in acrosomal reaction, it occurs after binding to the zona pellucida and it is induced by the zona proteins. Outer membrane of the acrosome fuses at many places with overlying cell membrane of the sperm head, and the fused membranes then rupture, producing multiple penetrations and perforation through which enzyme the hyaluronidase in the acrosome escapes and oozes out. So different enzymes which are released during the process of acrosome reaction are the hyaluronidase, the trypsin, the acrosine, and the progesterone like substances. So hyaluronidase needed to assist in penetration of the corona radiata barrier. The trypsin-like substance needed for digestion of the zona pellucida, and acrosine also need to help the sperm across the zona pellucida, and the progesterone, which is present in follicular fluid, seems to stimulate the acrosomal reactions. So different phases of the fertilization, you understand, this is the sperm, this is the oocyte, secondary oocyte, and it is arrested at metaphase of second meiotic division, and here the first polar body is present. This green color shows the zona pellucida, and surrounding the zona pellucida, these are cells forming the corona radiata. So first phase of the fertilization is passage through the corona radiata. These are different sperms which are passing through the corona radiata, and they starts to attach to the zona pellucida. So hyaluronidase from the acrosome helps in penetration. The sperm tail motility and table mucosal enzymes also help in the penetration. So when they penetrate, then penetration of the zona pellucida is facilitated by the acrosine, neuroaminidases and esterases, and then zona reaction takes place. During this zona reaction, the lysosomal enzymes of the cortical granules of the oocyte cell membrane are released, and these cortical granules alter the characteristics of the zona pellucida so that the zona pellucida become impermeable to other spermatozoa. So fusion of the plasma membrane and secondary oocyte and sperm, there is completion of second meiotic division of oocyte and formation of the female pronucleus. At the same time, male pronucleus is also formed, and fusion of these nuclei to form the zygote. So secondary oocyte is usually fertilized within 12 hours after the fertilization. IVF, unfertilized in vitro fertilization, the unfertilized secondary oocytes dies within 12 to 24 hours. So different phases. First phase is penetration of the corona radiata. Second is penetration of the zona pellucida. Third phase is penetration of the oocyte cell membrane and formation of the zygote. About 200 to 500 million sperms which are deposited in female genital tract. Only 200 to 500 sperm reach the site of the fertilization, and only one of this sperm is marked for fertilization. So Allah Himself knows the secret of this super selection of the genetic material. So once they crosses through the zona pellucida, then they starts to attach to the zona pellucida. The acrosine causes lysis of the zona pellucida, thereby forming a path for the sperm to follow. Once the sperm touches the pronuclear membrane, it becomes fully attached to it and penetrate it rapidly. So at this time, as one sperm passes through zona pellucida, permeability of zona pellucida changes and zona pellucida become impermeable to other sperm. This is called the zona reaction. It is believed that the granules released from the secondary oocyte, these are granules which are released from the secondary oocytes after the attachment of the spermatozoa to the cell membrane of the oocyte. These are the cortical granules, and these granules contain the lysosomal enzymes. So these granules changing the characteristics of the zona pellucida. Here you can see the zona pellucida has integrins and lesions which are present, and the disintegrins, for these integrins are present on the cell membrane of the spermatozoa, and there is interaction between the integrin and disintegrin which causes attachment of the spermatozoa to the zona pellucida. So once it is attached, cortical granules are released. These cortical granules then changes the character of the zona pellucida. So further no attachment of these spermatozoa to the zona pellucida occur, and this process helps to prevent the process of the polyspermy. After passing through the zona pellucida, the sperms become attached to the cell membrane of the secondary oocyte, and then head and tail of the sperm enters the cytoplasm of the oocyte, leaving the sperm plasma membrane attached to plasma membrane of the oocyte. The secondary oocyte completes the second meiotic division, forming the mature ovum and the second polar body. The nucleus of the ovum is now known as the female pronucleus, and the second meiotic division is completed immediately after the entry of the spermatocyte into the cytoplasm of the oocyte, secondary oocytes. So once within the cytoplasm of the ovum, tail of the sperm degenerates and its head enlarges to form a vesicular nucleus called the male pronucleus. So morphologically, there is no difference between the male and the female pronuclei. Another important point to remember over here is that the pronuclei fusion as such do not occur. The two pronuclear envelopes disappear and two chromosomes group move together to assume a position of the first cleavage. And another important point over here is that before they arrange themselves and they align themselves at the position of the first cleavage, first they have to duplicate or replicate their amount of the DNA, so that after the first cleavage division, each cell receives the diploid number of chromosomes and normal the amount of the DNA. So this is the revision slide. Here you can see the this is the jelly coat, which is the zona pellucida, and the sperm's binding receptors are over here. This is the sperm head which is releasing, the acrosome is releasing is enzyme, and acrosome is now, because of the enzyme, there is the ligand binding over here, the integrins and disintegrins binding over here, and after this binding the spermatozoa is attached, and this spermatozoa then enters into the cytoplasm, excytoplasm, and as a result of the entry, the cortical granules are secreted, and which alter the characteristics of the zona pellucida. Another important thing you must remember, that in human beings no true zygote containing the membrane bounded nucleus is ever formed. So this is important point, that in human beings no true zygote is formed which is bounded by a nuclear envelope. So what are the results of the fertilization? Important viva question. That is the restoration of the diploid number of the chromosome, the species variation, the shuffling of the gene. The zygote contains a new combination of the chromosomes different from the parent, and alpha form the basis of the biparental inheritance and results in variation of the human species. So here are the different clinical correlates with this fertilization, the assisted reproductive techniques. So in assisted reproductive techniques, we have different techniques: the in vitro fertilization, the intracytoplasmic injection of the sperm, zygote in vitro fertilization, and gamete in vitro fertilization. So these are the different methods. All these are the different methods help to patient for the we having the problem of the infertility. For the fertilization, we have them with these methods. In zygote intrafallopian in vitro fertilization, the first of all, the female patient is given the gonadotrophin hormone to stimulate the process of the oogenesis, and just before the process of the ovulation, then the oocyte is taken, laparoscopically taken, and then grown into the culture medium, where the where the spermatocyte is then introduced into that culture media, and they are then the fertilization between sperms and oocyte takes place, and after the process of the fertilization, the zygote is taken and then it is implanted in the uterine cavity. And in this process, the gametes are formed in vitro, and after the formation of the gametes, they are transferred into the uterine cavity. In this process, ICSI, that is the cytoplasmic sperm injection. This process is usually help those patients having either oligospermia or azoospermia. So in this case, only one of the spermatozoa is taken and it is directly injected into the cytoplasm of the oocyte, after which the fertilization occur. So you must remember, in all of these processes there are high rates of the congenital anomalies, as well as rate of the congenital anomaly also increases with the age. Also there is a failure rate as well, and failure rate also increases with age. Another important thing, as we usually implant 6 to 8 zygote or 6 to 8 gametes into the fallopian, into the uterine cavity, so there are the chances of the multiple pregnancies as well. So with the help of the ART, there may be pre-selection of the embryo sex, primary sex ratio can be determined, cryopreservation of embryo or zygote can also take place for the next attempt of the either ICSI or ZIFT or GIFT. Another important thing you should understand is the surrogate mothers. So in process of the surrogate mother, actually the sperm and the oocytes are taken, the oocyte is taken from another female, and another patient is prepared for the process of the pregnancy, and she is given the hormone so that her endometrium shows changes and become receptive for the implantation. So another third person, a third female is used as a mother, where the zygote is implanted in her uterus and she is acting as a surrogate mother. So these are the different clinical correlates. Then first, cleavage occur, which are the series of the mitotic cell division of zygote to form the embryonic blastomeres. So important point, another you must remember, which is an important MCQ, that the first cleavage usually occur 30 hours after fertilization, and the 4 cell stage occurs usually 40 hours after the fertilization, and 12 to 16 cell stage usually occurs after the 4 days, approximately at the 4 days. So these all the days and the hours are MCQs and important from your paper point of view. Next, implantation, the process during which the blastocyst attach itself to the endometrium. So this is the morula stage. These are the different stages. First 2 cell stage, then 8 cell stage, and still this stage, at this stage the zona pellucida is present. Here the 12 to 32 cell stage, it is called the morula, which resembles mulberry, and you know over here you can see the zona pellucida. But at this stage, when a cystic space appear, and this cystic space is called the blastocystic cavity, and at this stage the embryo is called blastocyst. At this stage, zona pellucida disintegrate, which is called hatching, after which the implantation of the blastocyst into uterine endometrium takes place. So here you can see different phases. This is the release, so from here we start. This is the release of the secondary oocyte at the time of ovulation, and it is captured by the infundibulum, and usually ampullary part of the uterine tube, which is closest to the ovary, is the part where the fertilization takes place. So this is again your MCQ. After fertilization, you can see 2 cell stage, 4 cell stage, 8 cell stage, then 12 to 32 cell stage where it is called morula. When it is morula, then it at this stage it enters into the uterine cavity, and here it becomes the blastocyst, because some fluid enters into the from the uterine cavity into the morula, and a cystic cavity is developed, and the cystic cavity is called blastocystic cavity or blastocele, and at this stage the embryo is called the blastula or blastocyst. So this blastocyst then itself it attaches and it reorients itself in such a way that its embryonic pole faces the uterine endometrium, and it then attached to the endometrium, and then the process of the implantation takes place. So dear student, there is a video for fertilization over here, and I will give the link in description to this video. You can see this video on YouTube as well. And these are the references: the Langman Medical Embryology and the Developing Human by the Keith L. Moore and TV and Prasad. And thank you so much for your attention for the today's class, and inshallah we will meet in the next class. Till then, Allah Hafiz.